Hello, everyone. Thank you for coming today. And thanks to the organizers of this seminar series for giving us core directors the opportunity to tell you about our newest technologies and services. And I hope at the end of this talk, I will have a chance during the question and answer session to hear from you suggestions about, what kind of services and capabilities you would like us to add in the near future, things that you might need in your research, but you currently do not have access to. A little bit of background on our core. Keck is one of the oldest cores at Northwestern, established in 1998, and it has been a Lurie Cancer Center shared resource since 1999, and one of the Feinberg shared resources since 2012. Our mission is to support scientific research at Northwestern by providing high-end biophysical instrumentation, along with technical expertise and specialized services. And we are a multi-instrument user core. We have currently 21 active instruments, 20 of them located in Evanston, one in Chicago. And although our facility is old, our instruments are not. In fact, most of them are state-of-the-art. 9 instruments have been added to the core just in the last 40 years. Although there's some overlap, we can categorize our instrumentation based on the type of applications. In these categories, we have instruments for characterization of molecular interactions, some for characterization of biological and synthetic polymers and nanoparticles. We have preparative equipment and also gel blot plate imagers. And we are a walk-up facility, but also a service center. What I mean by that is our instruments are available for self-service. We have about 300 users authorized to use the instruments, and they can be reserved via Newport. The facility provides a series of supporting services, including training, assistance, calibrations, guidelines, protocols, and such. We also offer staff service on most of our instruments. You can submit the sample and the staff will run it for you after a consultation, so we can understand what's needed exactly. We take care of everything from experimental design, sample preparation, running the experiment, data analysis, and report. And then we also have a newer category of services, which we call integrated services. These are designed to leverage our instrumentation and technical expertise and address specific needs within the research community at Northwestern. They typically involve using multiple techniques, and they are performed exclusively by the staff. And in this way, we can optimize the workflow such that they are performed with minimal sample consumption, time, and cost. And since these are newer, and in my opinion, more relevant for this audience, I'm gonna spend more time today telling you about how this works and what they are. But if there's anything in particular about any of our instruments that you wish to discuss, please feel free to ask me during the question and answer session. So the first example of integrated service we offer is called protein characterization and quality control. And we use a combination of spectroscopic CD, fluorescence, absorbance, static and dynamic light scattering, separative techniques, calorimetry, in order to interrogate solubility, stability, purity, conformational heterogeneity, and higher order structure in proteins. Typical applications for the service are structural activity studies, studying and understanding effects of mutations, temperature, pH, storage conditions, cofactors, etc. And also quality assessment of and optimization of recombinant proteins. So with recombinant proteins, when they're used in a biophysical, certain biochemical, certain types of high throughput assays, we want to make sure that the protein is high quality. This is required for accurate and reproducible data. So by that we mean the protein is pure, the concentration is accurately measured, and the protein is in its native active state under the conditions in our experiment. And a lot of effort and care is put into making sure that conditions 1 and 2 are met. But oftentimes condition number 3 is overlooked. And this is particularly the case when we buy proteins from commercial manufacturers. They will typically guarantee the concentration and purity, but there's no way they can guarantee that the protein they provide is going to be active exactly under the conditions you need in your assay. So I'll give you an example from our own lab. We are working with a group here at Feinberg in a project, it was a drug discovery project at the lead optimization stage. So we were given this protein that they purchased from a commercial manufacturer. It came with a quality certificate stating that the protein is 95% pure. That was based on STS gel analysis. And based on 826 to 80 absorbance, it was also free of contamination in nucleic acids. And the concentration was stated at 1 mg per ml. But yet in our assays, the protein failed to bind. We had very, very low activity. We first suspected, the compounds, but luckily we had some controls. And when we ran them, we realized that the protein is less than 10% active. So when we looked at the causes of this, we checked by UV absorbance. The concentration and purity was exactly as specified on the certificate. We then ran dynamic light scattering, which showed that there's no large aggregates or degradation in the protein. However, the close inspection, the polydispersity index and the hydrodynamic radius in the light scattering were higher than expected. One explanation for that is that the protein was unfolded, but we checked which circular dichroism and the secondary structure was present and the amount and type expected. And then when we look at the quaternary structure, via analytical size exclusion with light scattering, we found that only 8% of the protein was the active monomer. The rest of it was a combination of higher order oligomers from like a 30-mer to a tetramer. Of course, you will not see this in an STS gel because the quaternary structure is not preserved. And also, you probably won't be able to eliminate these oligomers by spinning down the protein or filtration. So understanding the problem sometimes helps because some of these things could be fixed. So this is just an example. We also found situations where the protein is unfolded or other problems with them. So if you suspect that the protein is to blame, your assay and controls do not work as expected, you can request this type of service from us. We have a form on our website where you can submit a request. Once we schedule you, you can send a sample via the intercampus courier, and you have results in maximum 48 hours. So another type of service we offer is the biophysical characterization of drug-target interactions. This is meant to support drug discovery efforts at Northwestern, and it's utilized at the lead discovery stage in the process, and it consists in secondary screening via orthogonal techniques confirming the hits found in high throughput assays, ranking of these hits, lead identification, and then thermodynamic and kinetic profiling of the lead target interactions. And for this service, we use a combination of techniques, most often surface plasmon resonance, biolayer interferometry. They both are optical sensing techniques, which record the association and dissociation of molecules in real time and provide affinity and kinetics. And isothermal titration calorimetry, which records detects enthalpy of binding in titration. And from that, you can get, again, affinity, stoichiometry, and a complete thermodynamic characterization of the interaction. The advantages of using these techniques as secondary screening methods are several. First of all, as I said, they are orthogonal to what's typically used in high throughput screens. So you provide a confirmation by a different method. Moreover, they are much more precise in terms of measuring affinity. They are also label free. They can be performed under physiologically relevant conditions. And they can measure both weak and very strong affinities. They have very little limitation in terms of sizes of compounds that can be measured. And perhaps equally important, they provide information beyond affinity. And by that, I mean kinetic and thermodynamic characterization of the interactions. So from thermodynamic characterization, we can dissect the binding free energy into its enthalpic and entropic components represented. So free energy, it's blue bars here, and then enthalpy green and entropy red. So in this figure, we are looking at 3 different compounds binding a target with the same exact affinity, but yet they have very different thermodynamic profiles. And because we know what contributes to favorable enthalpy and entropy, we can understand why these molecules bind. What are the interactions that favor binding? And when we combine this with structural information, they provide a picture of the molecular interface, and we can validate the assumptions made about the interactions that take place. So this type of information is very valuable in the lead optimization stage, where it can validate data from molecular dynamic simulation and correlate with structural information available. Similarly, from kinetics, we can understand how the molecules bind. If we look at an example of 4 compounds binding the same target with, again, the same exact affinity, we can see that they actually bind in very different ways. So the compound with the red trace binds with fast affinity, fast kinetics. It forms the complex really rapidly, but also the complex is short-lived. For the green and dark blue, they form the complex slower, but once formed, this complex is very stable. So the kinetic profiling is being increasingly used in drug design, especially in cases where the interaction with target is complex. And one such example are the PROTAC, design of the PROTAC compounds. So These are compounds that intend to target protein for degradation, and the way this process works is schematized here. So the idea is to induce the proximity between a protein of interest and a ligase, a specific ligase. Once this complex forms, the ubiquitin charge E2 enzyme can transfer the ubiquitin to the protein of interest and initiate the degradation via the ubiquitin proteasome system. But designing this compound is not trivial because normally you would link a specific target for the protein of interest with a target for the ligase. And one of the things that needs to be assured is that this particular complex forms with very high specificity. The complex between the drug and the protein of interest. So the PROTAC compound can bind to both the ligase and the protein of interest, forming these binary complexes. But from studies that have been published so far, looking at why some PROTAC compounds are more efficient than others and what happens at the molecular level, it is clear that this complex is very important. It's both its affinity and stability are critically important to make the process effective. So therefore, this particular complex needs to be favored and needs to have enough stability and affinity for this obviously one of the factors that helps a PROTAC to be effective. So we had the opportunity to participate in this study, which aimed to identify PROTAC degrader for the IDO1 protein. Most of this work was done in Derek Wainwright lab and at ChemCore, but also in collaboration with many other groups here at the Cancer Center. IDO1 is a valuable target in cancer therapy. It is a potent inhibitor of the anti-tumor immune response. It is regulated in many human cancers. And although some inhibitors of its enzymatic function have been developed, they do not appear to improve the cancer patient outcome. So therefore, targeting this protein for degradation might be a more effective approach. So in the study, a library of PROTAC candidates were developed at ChemCore, starting from a crystal structure of IDO1 with an enzymatic inhibitor bound to its active site. And the library was consisted in linking this inhibitor here for IDO1 with various types of ligand for the E3 ligase and with linkers of various lengths and composition. And after 2 rounds of screening in U87 cells simulated with interferon gamma, they found 1 out of 100 compounds that was efficient in degrading IDO1 in a dose-dependent manner. So in Keck, we looked at the complexes formed by this PROTAC compound with both the IDO1 enzyme and the cereblon ligase. These are biolayer interferometry, BLI, and calorimetry data. And we characterize both the kinetics and thermodynamics of these complexes. And we are also able to detect and measure the ternary complex formed by the two proteins and mediated specifically, as we run this control here, by the PROTAC compound. We saw that indeed this complex has higher affinity and stability than either of the binary complexes. So we developed this framework. In fact, we found that biolayer interferometry is ideally suited for characterizing ternary complexes because it's sensitive to size, not just to mass and concentration. So therefore, a protein-protein complex can be easily distinguished based on the magnitude of the signal from a binary complex, which is just protein with the compound. That is not so easy to do with either techniques such as ITC and even SPR. You will need some controls in order to make sure you're looking at the ternary complex and not the binary ones. So right now we're starting a project where we're going to follow up on this. Is the compound is currently being optimized, so we're going to measure more interactions with hopefully better PROTACs. This was just one example. We applied the service for many other projects. Some of them are shown here, and we can customize, what combination of techniques and what exactly we need to be looking for in drug development studies. And I just want to reemphasize that this service is exclusively available to Northwestern users. This is not available for external users, and it's prioritized for cancer center members. So they have priority in schedule, they have discounted rates and assistance with preparation of data for publication and grants. Also, this type of work is eligible for the H grants, that H Foundation grants that are distributed by the Cancer Center, also CDC funding. So if you have any, interest in applying for such funding, just let me know, and I'd be happy to provide, letters of support and information, budget, and everything that's needed. So I'd like to move on to our next type of service, which is also our newest one. It's for separation and characterization of biological, synthetic polymers, and nanoparticles. And it's based on a multi-instrument system that was recently funded by NIH. The heart of the system is this separation unit based on a new method called field flow fractionation, or FFF. And then this is interfaced with a multitude of detectors, absorbance, static, dynamic light scattering, refractive index. So how does this separation work? I have just the schematics here. It's a microfluidic channel, and you inject a sample in the channel. It is concentrated briefly on the bottom of the channel, and then it's allowed to relax under a cross field. The smaller molecules diffuse faster, so they will reach the top of the channel first, and then the larger molecules will stay close to the bottom. And we apply this parabolic flow field and in in this way, the molecules that are close to the top of the channel will exit the channel first, and the ones on the bottom will exit later. So that's how separation is achieved. But the power of this method is unparalleled because we can separate anything from small polymers to cytokines to nanobodies, proteins, protein aggregates, viruses, all sorts of nanoparticles, and all the way to exosomes. So, moreover, by tuning in the parameters of the field, you can adjust the separation, so you can perform both coarse and fine separations. So this is an example of separating proteins from whole serum. In a 1 hour run, you can see that distinct peaks are observed for the IgG, HSA, and all sorts of lipoproteins present. The second strength of the system is its capability for characterizing multiple attributes of the samples. So, for example, for liquid nanoparticles, we can get all this information from one run. This is possible, of course, with other techniques, by separate measurements. But here, based on the fact that we use not just individual detectors, but information that comes from combining two detectors, it is possible to get all this information. So, for example, for liquid nanoparticles with RNA, if you look, these are 3 types of nanoparticles. The red one is with low RNA. Empty particles that have no RNA are blue, and green are, red are with high RNA content, and green are with low. So if you just look at the separation itself or the light scattering signal that's provided by one detector, there's actually not a good way to tell them apart. But if you look at the combination of the hydrodynamic and the gyration ratio that's coming from 2 detectors, you can immediately identify the nanoparticles that are with high RNA encapsulated from the low. And that's because there's a specific value of this ratio that is for hard nanoparticles versus empty nanoparticles. Hard, I mean nanoparticles full of material. So again, this is just one example. You can also look at the efficiency of encapsulation and many other attributes. And then lastly, the technique is both analytical and preparative. So that means you can collect these fractions and utilize them either for downstream applications, and that's important for nanoparticles intended for therapeutic purposes, where having homogeneous populations is required. And also you can collect them and analyze by different methods. So one such example comes from this article that actually reported for the first time the identification of exomeres. I'm not sure if you're familiar with this, but basically they initially attempted to use FFF in order to separate exosomes that were seen as being very polydispersed in TEM and also in nanoparticle tracking analysis assays. So they applied FFF to this. They were successfully and easily separated 3 distinct classes of such particles. and they termed them XOL, large exosomes, XOS, small, and then something they have not seen before, some particles with different morphology, smaller than 50 nanometers, and they call those exomeres. So this was from one type, the exosomes derives from one type of cultured cells. And the assay was very reproducible. Every single time they separated these three categories. And then they got 20 more cell lines from collaborators across the world, and the same distribution was seen in all of those populations of exosomes. And since I just told you that it is possible to collect these samples, they did just that. They collected from every single type of cells, and they subjected this to a very detailed analysis. And they found out that exomeres in particular, but also exosome small and large, have very unique protein, lipid, DNA, RNA, and also I think and glycosylation profiles. So this was, I mean, there's a lot in that article that needs attention, but this is not, you know, the time to discuss everything they found. I just want to say that this is their first paragraph in the discussion of the article, and it just clearly states how this method is superior to everything else they tried before in order to separate exosomes. You can efficiently and reliably resolve these populations in a single run. And if I got you confused with the terminology, AF4 is just a subtype of FFF. It stands for asymmetric flow FFF. And this is exactly the technique we have in our system. And in fact, we have this exact same setup that they used in this article. But our system is new. We haven't run much on it so far. Just some proteins, protein aggregates, and some lipid nanoparticles. So we invite, any collaborations, interesting ideas, samples that are new. We are looking to kind of take advantage of the capabilities of this system. So if you have any ideas, for maybe you need some preliminary data for a grant application or for a new type of application with the system, please let us know. The only samples that do not work here are very elongated samples, such as nanofibers or carbon nanotubes, or flat samples that are like sheets and things like that. But everything else that has dimensions in this range would work well. So this is my last slide in terms of services. Just want to introduce you to our small team. Amy Rosenzweig has been the faculty director of our facility since 2011. I've been managing director since 2006. Jimmy Casey is our assistant manager. He has been with core facility since 2017, and David is newer. He just joined last year. I also want to acknowledge our sources of funding. We receive operational funds from Lurie Cancer Center and Feinberg School of Medicine, and our instrumentation funds come from the NIH and the Office of Research. This is how our contact information. There's a lot of notes, protocols, information about our instruments on our website. Please visit that. You can also find us with this new Cancer Center Core Navigator that was, I think, just introduced today. And with that, I'd like to again hear from you, specific questions or suggestions you have for us. Yes. It's a good question. We have separate cells for analytical runs and preparative. So I'm not sure which one. So are you concerned about the amount of sample that is needed for an analytical run, or yes? I don't think I heard you one. So the injection volumes could range from 10 microliters to 500 microliters. Concentration required are varying because much larger samples require less material than smaller ones. So it really depends a lot on the sample. How fast it is? So usually, so how fast is the separation? So the runs range from 30 minutes to maybe 90 minutes, depending on how complex the mixture is. You will need to keep the cross flow longer if you have a sample where, there's a very heterogeneous population with very small and very large things. If you want fine separation, that's doable in a shorter time. Okay. You had a question too? Mm-hmm. Right. It is. Yeah, but I mean, it is better in terms of preparative capabilities than size exclusion, but it's not like, the columns that you typically use for preparative. You can put maybe a maximum of 5 mg of protein in one run. So it depends on your scale. It can be, if you're looking at large scale, small scale, or medium, but you cannot do large scale separations. You will need to do a lot of runs because every single run can handle just 5 mg of protein. But it is, we run it with some protein samples, and it's for analytical research required a lot less than SEC, maybe 10 times less material. So that's really, yes. I need to come closer again. Mm-hmm. Ultracentrifugation, I guess. Yeah, we have that too. Yeah. This one, we do have what's called a dilution control module in it. It's a bit hard to explain, but you could apply another flow field that essentially would limit the range where the particles are collected from. So you could attempt some concentrations too with this, but I guess it's not as much as it will be with the ultracentrifuge.